now we're getting into something which I think our listeners are going to find really fascinating, mind-boggling, and honestly, it's very unique and inspiring for me. And we are talking about your rat yoga experiments. When I tell people this, they're like, "Wait, can rat do yoga?" And I tell them, yes, there is a school researcher who shows us the same forces that we experience in our daily lives. Like for what you would experience in yoga or physiotherapy or even gentle movement or stretching, they can actually be applied in a controlled lab setting. And she's seen it how it can influence cancer. So that's even more fun. And the implications are a lot serious for this, not just for understanding cancer biology, but also for imagining these new movement-based complementary therapies. So let's dive into this topic and hear about the experiments from you. So can you please start by describing some of your key cell or animal studies where physical stretching influenced tumor growth, and what kinds of physical stretching or movements did you test? How you tested them in an ethical constraints, and what were the primary outcomes that you observed?
Well, thank you. So we were originally interested, my lab was originally interested in looking at the effect of gentle stretching on inflammation, chronic inflammation in animal models of subcutaneous inflammation. So we induced a very small inflammatory lesion, chronic inflammatory lesion in the back of rats at the time. We then moved on to mice. And then in an animal, what you can do is you gently lift the animal, the rat, by the tail, and you allow the animal to grasp onto a bar. And then the animal spontaneously sort of stretches their whole body. They can learn how to do this. We do it very gently. Let the animal get used to it. And they can gradually increase the length of time that they can do this to approximately 10 minutes. And we do this twice a day. And what we found is that the growth of the inflammatory lesion was reduced over the course of 6 weeks in the animals that had been stretched. And so we were just curious, since there is a link between chronic inflammation and cancer, we were curious about whether a cancer model, what would happen in a cancer model. So the first experiment, we were very surprised that it worked. We injected some cancer cells in the same location as the carrageenan in the back, in a subcutaneous space between the thoracolumbar fascia and the skin, subcutaneously stretch the animals, and we saw, to our real great surprise, really, that the growth of these tumors was reduced by 50%. And then we wanted to make sure that this was real, so we've replicated this in many different cancer models, and we always find this pretty much the same result. We also wanted to see if you could stretch the animal passively, because there's a lot that happens when you hold the animal by the tail. They're stressed, they have muscle activity, there's all kinds of things. So we anesthetize the animal and we'll manually just gently stretch the distance between the shoulders and the hip to replicate what happens during the spontaneous stretching. We saw the same thing, very similar reduction in the growth of the cancer. We also have a very interesting model where the animals stretch spontaneously by simply putting their food and water in a cage high up, versus the control is simply the food and the water on the ground of the cage. And we see very, very similar results, again, about approximately 50% reduction in the growth of the tumors. So this is something that we feel is robust result. There's something about the application of this mechanical force which is beneficial, which is very sort of surprising in a way, because a lot of the basic research, such as what I believe you're doing in your lab, right, where you're looking at mechanical tension in the form of forces applied by myofibroblasts, for example, in tissue. When you find that you have a lot of myofibroblasts in the tissues, there's traction around the tumor that makes the tumor worse. So increases the growth. And so we were really baffled by this result. We looked at the relationship of the stretching to collagen depositions, for example, myofibroblasts. We did not see any indication that there was a reduction in that. And so we are very interested now is looking at the effect of the stretching on the tumor cells themselves. And so we have developed in vitro models looking at like in tumor spheroids, for example, and looking at changes, for example, in RNA expression in these tumors. And we're sort of in the middle of trying to understand uh, the effects of stretching, because it's very surprising to us, but it's real.
I was wondering about the precautions that you would take in this type of research. And like you had mentioned, when we apply forces, it can have several effects. So one concern is that when applying forces right on a tumor or over it, or might it dislodge the malignant cells and promote metastasis? And the second concern that you had already pointed out is when we apply forces in and around the tumor, there are some local effects that happen. The mechanical forces inadvertently, they promote tumor progression through mechanotransduction. So how would you separate these local versus the systemic effects? And how do you control for the dose, frequency, and duration so that, like you said, the mechanical forces that you're applying by stretching are beneficial?
Yeah, very important. We were concerned about the metastatic risk from the start. And this is one of the reasons why we've moved very slowly in our preclinical models to make sure that there's no additional risk posed by the stretching. So this was challenging because the model that we were using, the initial model that we used back when I used to be in Boston, was a P53, P10 double null memory cancer. And within the course of the experiment that we were looking at a 3-week span, and the tumors by that point grow to the point where at the end of 3 weeks you have to euthanize the animals because they can ulcerate and it becomes uncomfortable for the animals. So we have to euthanize. They did not have metastases at that point, so we were not able to look at that. So after I moved to NIH, we moved to a polyoma middle T model, where the tumors are more aggressive and they do metastasize. When we first look at we looked at both an orthotopic and a transgenic model, and in both models, the effect of the stretching was just as effective as in the 53 P10, very similar results. However, some animals develop metastases and others not. It was variable. We did not see a difference between the stretch and the no stretch group with respect to the metastases. But because there were so few animals that developed metastases within that time frame, we were not able to do proper statistics on it. There was just not enough metastasis. So just recently, we've now moved to a new model that has more aggressive, and where pretty much all the animals develop metastases. And we're in the middle of doing these analyses. But so far, what we're finding is that there's certainly not more metastases. If anything, the stretching reduces the number of metastases. So as far as that is concerned, I think we are very encouraged by these latest results. And the amount of stretching that we do is, first of all, very, very gentle. It's not the same thing as when you apply pressure, like during massage, for example. Some people have seen that, right? When people do like sentinel node biopsies, for example, where you apply a direct mechanical pressure, and you can see that there are cells on the tumor. There are cells that migrate to the lymph node. That's well documented.
Mhm.
Here, the stretching is applied not via pressure, but it's more like in yoga, for example. It's much more gentle. So we're thinking that maybe that's the reason why the metastases are not increased, but it's definitely a concern. I think that we feel that our animal experiments right now are definitely reassuring in that area. But importantly, as you mentioned, understanding how to apply the correct dose of stretching is going to be really, really key, not just for avoiding metastases, but also for avoiding excessive stretching of the primary tumor.
Mm.
Um, So right now, we think that the correct amount of shear strain applied is on the order of 25% shear, which is, when you think about 100% shear, if you have two tissues that are like this, right, one next to the other, 100% shear would be all the way out to here, right? If you were to stretch where you have two pieces of something, right, and 100% shear would be all the way to here. 20 to 25% shear would be about this much,
Mhm. Okay.
right? And when you look at, and we've done a few studies, not nothing systematic, but just to try to get an idea of the amount of stretching that occurs during in certain areas of the fascia that you can measure. For example, in the thoracolumbar fascia, that when movements that patients do spontaneously, that's about a good range. 20% shear typically would be the kind of stretching that people would do, like say if you're reaching up for a shelf, like our animals that are reaching up for their water bottle.
Mhm.
It's not excessive. So this is where we are also encouraged, the kinds of stretching that people do with everyday living. Like say, for example, when you're reaching up and you want to reach something high, would be about the right range that would not likely to be injurious and probably would be more in the beneficial area.
Okay. Have you also tested when to start stretching, for example, before or after the tumor formation to explore the prevention or versus therapy effects?
Yeah.
Um,
From the beginning, yeah, good question. We never thought of stretching as a treatment, right, for cancer, clearly.
yes.
It's more preventive.
Mhm.
Uh, the tumors that we use in our models are tiny. They're very, very small, the order of, millimeters. So they are the stretching in our experiments so far are we start stretching the animals as soon as the tumor, the same day that we inject them. Uh, and uh, and then in the transgenic model, we start stretching the animals right after weaning, and the tumors develop spontaneously. And we find that the stretching prevents the development of the tumor. So again, we really are thinking of this more as a prevention, either primary prevention, uh, to so that people stay healthy and don't develop cancer in the first place,
Mhm.
or secondary prevention, say somebody has had already treatment for cancer and want to avoid a recurrence,
Mhm.
people who are at risk for a recurrence. There's also a really interesting possibility here now there was a clinical trial recently that was a pragmatic trial looking at women with stage 0 carcinoma in situ, where there was a they were randomized to either treatment or just a watching and waiting. That would be also an important, and they found that the watching and waiting was not
Mhm.
inferior to treatment. So this is quite an interesting area now that would open up the possibility of doing a clinical trial of stretching in the context of observation for stage 0 cancer. This is still a little bit. There's 2 other clinical trials right now that are in progress and it'll be interesting to see if they find the same results. But if they do, I think this will open up the opportunity of studying interventions such as not just stretching, but other types of interventions, lifestyle interventions, for example,
Mhm.
with in these women with minimally invasive cancers.
I was actually also wondering about the high risk environments. For example, people who have germline mutations like BRCA1, and you've
Yeah,
already mentioned the P53 null mice, and also like people who are more exposed to carcinogens, or like, for example, obese mice. Do you
yeah.
think your model would be applicable in those?
Absolutely. So again, this would be primary prevention, right? This would be lifestyle. We know already that having a healthy lifestyle protects against the development of cancer over the lifetime, but we don't really understand exactly the mechanism of this, right? It's probably multifactorial if you have a healthy lifestyle. And again, we're going back to the principles of Ayurveda. You pay attention to diet, you pay attention to movement, exercise, and also you pay attention to the mind and the, for example, healthy sleep is very important, that kind of thing. So it's probably a multifactorial effect. but it would be really important to understand the components,
Mm,
the individual components of these sort of types of intervention.
mm.
Uh, but ultimately, you probably want to apply them as a whole. Why not? If you want to have a lifestyle intervention, it would make sense that you would address the nutritional, the physical, and the psychological components together.
Yes. I was curious to know about the mechanisms behind this phenomenon. Um, and I'm wondering, like, the tumor size would be really small to do any kind of analysis, I'm assuming. Um, but I was still curious if you were able to harvest the tumors and if you were able to look at some of the mechanistic markers like inflammation or metabolomics or angiogenesis on some of the invasion markers and also stiffness and mechanobiology and mechanosensing markers.
Yeah, absolutely. Yeah. So we are in the current in a process of doing some spatial transcriptomics on the tissues harvested tumors after they have been stretched versus non-stretched. And I don't have the results of this yet, but I think it'll be very important to look at all the various cell populations because, as we know, right, as you mentioned at the beginning of the podcast, it's an ecosystem, right? There are immune cells that are interacting with the tumor all the time. We have blood vessels. There is the extracellular matrix. All of that may change as a result of stretching. Now, we also have a simpler model where we take the cancer cells and we grow them in a collagen gel, and it's a thick, collagen gel that we're able to stretch, and we mimic the stretching conditions in this. And here, all you have is tumor cells. There's no immune cells. There's nothing else. And what we find is this the spheroid, the cancer tumor spheroids also reduced in the growth in the spheroids. So we know that at least part of the effect of stretching is independent of the immune system. Not to say that there might not also be, it may be that part of the stretching is also having an effect on the types of immune cells that are able to now get into the tumor and maybe have cytotoxic effects, and we're very interested in that as well. But the results in the spheroids are very provocative because we are able to do, I've mentioned, looking at atomic force microscopy, for example, we're able to measure the stiffness, not of the tumor cells inside the tumor itself, but also on the periphery. And we also measured the tumor-associated collagen signatures, like you mentioned. We see that they are different. So stretching these both the tumors in vivo as well as in vitro changes the organization of the collagen fibers surrounding the tumor, and we think maybe it makes it less advantageous for the tumor cells to start invading. It slows down the invasion,
Mhm.
so that it may be that then it gives a chance for the immune system to go and do its job. So it could be that there's biomechanical effects and also other types of effects such as immune.
Yeah, I think it's quite intuitive that the stretching would reorganize the collagen and rearrange it in a manner that it's not um, um, that it doesn't allow invasion. Yeah, so that's
Yeah,
quite intuitive and it's kind of fun to understand.
discourages it doesn't really it's not like chemotherapy, right? That's going to just go and kill the cells.
Yes.
The cells are still proliferating, they're still growing, the tumor, but they're not growing as fast. And so you also that we have defenses, in our immune system against this type of thing, and maybe it just gives a chance to the immune system for going in and recognizing these abnormal cells that we know our T cells are able to do. It's just that when the tumor is growing too fast, then the immune system is overwhelmed.
Yes. Yes. Yes, that's right. Um, what do you think, what kind of data or validation would be needed to move this type of a study towards clinical trials? I know there's a lot to do right now, but only if you're
Yeah,
comfortable answering this.
yeah. Well, I think we're close. I mean, I think that determining, I think the most important thing right now would be to determine a protocol. that could be done with fidelity and reproducibility in order to have as measurable a dose you can. I mean, you're not going to be able to determine with exact precision the amount of stretching that somebody would do during yoga, for example, because we still have, everybody is different, right? Some people have, as we going back to the example of the rubber band, some people's connective tissues are more stiff, other people are less stiff. You can't just say we're going to, hang everybody a weight off your arm or something like that to stretch. That wouldn't work, right? People have to learn, and we think an important part of this is what we call proprioception, which is that people have to sense and feel what's going on, like that, like what you were talking about when you stretch your muscles after exercising and you feel better. Part of that is you apply stretching in a way that feels good to you. And may be different. The amount of stretching that is appropriate for one person may be different for another person. And how do you determine that? And so, that's going to be interesting. It's an interesting problem. I think that the most important thing is to not stretch too much. So I think my sense would be to err on the side of not enough, perhaps, or stretching very gently. The worst thing that can do is that you don't stretch enough, but stretching too much I think, could have a sort of a risk of first of all injury, and secondly may have detrimental effects. So I think that's going to be the challenge. It's going to be to determine the dose and how to administer this dose in a way that's safe.
The last section has some fun science questions for you that I'm really excited about. If we put on our researcher hats for a moment, I'd love to hear your thoughts on how you would design lab experiments to test the biological impact of the following modalities. Either it can be in general or in the context of cancer, it's up to you. So the first is acupuncture, the second is massage, and the third is sound therapy or mantra therapy. So yeah, let's start with acupuncture. There's absolutely no right or wrong here. I'm just excited to hear your thinking as a scientist, exploring these ideas in a playful, experimental way. That's it.
Sure. Well, you can imagine this is something I think about a lot. Maybe not the sound one. I'd have to think about that one, but that's an interesting one too, because sounds are mechanical waves. So I mean, and I spent a lot of work in my lab looking at ultrasound, so it's not that far away. But anyway, let's go back to acupuncture. One of the areas that I've been personally very interested in is the mechanical interaction of the needle with the tissues. So we usually think of, when we do acupuncture, we insert the needle, but we don't just insert the needle. We also manipulate the needle, typically a combination of movement up and down and twisting. And in my lab, we developed methods that was a long time ago, to measure the amount of force that the needle exerts. Very, very small amounts of force. And these are torques in the micronewton meters. I mean, just very, very small amounts of force. But I think that since then, a lot of work has been done to understand the role of the interstitium in different aspects of inflammation, for example. So the experiment I would love to do is to combine acupuncture with sort of new types of techniques that look at a fluid flow in the tissue in response to acupuncture. So recently, there's been some nice papers on the movement of fluorescein dyes through the interstitium. It would be really interesting, I think, to combine that with acupuncture. So that was your first question, was acupuncture. So off the top of my head, this was an experiment I'd like to do. And then you said massage. Well, here I have some help. We have a wonderful program that NCTIH has been funding for this several years now, called force-based manipulation. And these are research networks that bring together all different types of scientists that look at both the effect of the biomechanics on the tissue, but also on the nervous system. and how does applying a mechanical force have cascades of effects that can eventually lead to, in this case, they were very interested in pain, but I think it could be applied to any type of
Mhm.
types of tissue pathology by bringing together these sort of research networks. And so this force-based manipulation group has been fantastic in sort of coming up with really interesting experiments. So I would really encourage you to check out their website. It's called Neurocircuitry of Force-Based Manipulation.
Yeah.
Uh, and then and then sound. Uh, Boy, that is a really interesting one. Um, I think that we all know, right, that a vibration,
Mm.
right, caused by different types of acoustic uh, sound applied to the surface of the body can be measured. And ultrasound is a great example of that, where we can calculate the effects of the vibration. There's a really, really interesting technique called shear wave elastography. I don't know if you've come across it. You send an ultrasound pulse through the tissues, and then it generates these waves of shear waves through the tissue, and then you can use that to calculate a strain map
Mm.
and a stiffness map in
Mhm.
the tissue. Is fantastic technique. So that would kind of be my wish. I don't have one of those machines in my lab, but at the moment, but these are fantastic instruments that can really help understand this whole thing about the relationship of the mechanical input to the tissue response.
Mm. Yeah, interesting. Coming to those subtler aspects that you mentioned, like the sound waves and energy, we know that yoga is, or stretching is a multi-dimensional practice. It involves not only the stretching aspect, but also the breath, the mind, energy, um, that connection. So I'm curious how we might begin to scientifically study these more subtle aspects like breath work, which is also pranayam, or meditation, or energy flow through a scientific lens, and what kinds of methodologies or frameworks might we need?
Well, breathing is super. I mean, talk about breathing, even at its most crudest aspects, is essentially applying a mechanical force through the movement of the diaphragm, right? That gets transmitted to the internal organs. And it's one of my favorite things that I keep reminding people. If you wanted to do and find out, ask somebody who does yoga, is it possible that how you breathe influences your digestion, for example? I think a lot of people would say, well, probably, right? If you do a PubMed search for anything respiratory and digestive, you find nothing.
Yeah,
That's because,
nothing.
again, going back to what originally the concept of whole person health, your lungs and your gut are not disconnected. They are connected via the diaphragm in a lot of other ways. But
Mhm.
there's a mechanical relationship between the movements of the chest and what goes on in the abdomen, which we need to understand that better. And one of the ways that we should start with is to really measure the mechanical forces applied during breathing. I mean, this is fundamental. And how does that affect the movement and of the liver, the stomach, the colon, all these organs that are in our abdomen. So I would say that start there, a very low hanging fruit. Very Now, when you're talking about more subtle effects of breathing, we know that different breathing techniques that are done that are in conjunction with, for example, mindfulness, right? Of being aware of the breath, of being aware of there's different techniques of breathing through, one side of the nose, the other side of the nose, or sending the breath to different parts of the body. I mean, this really gets into a very important concept, which is called interoception,
Mhm.
the ability of our body, of to sense, interpret, and regulate the signals, back and forth signals to the internal organs in the through the nervous system. This is an area also that is very much of interest to NCCIH at the moment, where we really need to understand these pathways, and breathing is a very important function that is very much goes hand in hand with a lot of sort of interoceptive awareness. And in cultivating interoceptive awareness related to breathing is very linked to the function of the autonomic nervous system. So there's a lot there to explore related to some of these, therapies. Yeah.
I agree with you. Um, if you have no restrictions of funding, what are some of your dream experiments that you'll conduct
You mean in my lab?
in your lab?
Um,
Or
well, I already mentioned I would love to have an ultrasound machine that can do shear wave elastography. I think that would be fantastic. I think the imaging, more and more, the biggest challenge, I think, is to relate to what's going on at the macroscopic level and what's going on at the microscopic level in the same experiment. Very difficult, right? To apply a force to, say, an intact, say animal or human, and then be able to measure the force and the displacement and all the different types of strains that are occurring, calculate, the stiffness map, the shear map, and at the same time be able to measure what's going on at the cellular level. Obviously, that there your limitations of what you can do in humans, but with this, because a lot of measurement, but is destructive, right? I mean, you have to open the tissues, and it's hard because at the cellular level, you're limited by the resolution of your machine and also on the depth of penetration. So, for example, optical imaging that you can do through the skin has a very low, you can only measure what's going on at the surface. Although that's beginning to change, I think that there are increasingly optical methods that can measure deeper into the subcutaneous tissue, where non-invasively, where you could all start to see what's going on in the cells at the same time that you're applying the force. So I think, to me, that's the challenge, is to combine these type of imaging and biomechanical measurements all in one experiment, where, and that, of course, you said no no limits in funding. I think that's but I know that at the National Institute for Biomedical Engineering and Biomedical Imaging, they're really, really interested in funding this type of research, and there's a lot of progress happening right now. My lab is actually collaborating with intramural investigators there at NIBIB, and we're going to be working towards doing some of those complex experiments. But it really needs to be all put together in one. It's not easy.
yeah, that's a
But,
dream of all mechanobiologists.
yeah.
So with this, I think we've come to the end of the podcast. And I agree there's a lot of untapped potential in this field, and we're just scratching the surface, and new interesting things are unraveling. So it's very interesting. Your educational and research journey has been very unique. It has been very diverse, and your work has beautifully bridged disciplines that we rarely speak about together. For example, neurology and connective tissue, biomechanics, acupuncture, integrative medicine. It's a lot. So, could you please begin by sharing what led you to this journey and what drew you as a physician scientist into this fascinating intersection of these disciplines, and looking back, how has the journey been so far, and also what message would you like to give to the next generation of researchers and enthusiasts like myself, to inspire them and explore these holistic interventions as something which is legitimate and important areas of research.
Well, thank you. I love this question. Well, first of all, what drove me there was a little frustration. I was actually a practicing physician, and I felt limited by really having not a lot of options to offer patients. Pretty much all options for treatment were all, pharmacological. I felt that there could be more, and that's how I became interested in acupuncture as a modality, and ended up studying it, which got me to being very curious about what happens during acupuncture. When we twisted the needles, I could tell that there was something mechanically going on, but didn't know what it was. And then our first instrument was designing a little robot that inserted the needle and manipulated the needle and measured the forces. And that's how I got interested in that. That was kind of set me on a path to research in this area. And I would say the most important thing to encourage is to follow your curiosity. I think this is what is the most fun thing about science, is to follow the questions that intrigue you, even if it's not clear what the path is. Sometimes some of the most, and not be afraid to tackle questions that are a little bit outside the mainstream. I would say don't go too far outside the mainstream, because you got to be able to connect your question to what we already know, but just be far enough that you really stretch the limits of knowledge. I think is a very good thing. And so this is where I think devising research questions inspired by traditional healing traditions like Ayurveda is a wonderful thing, because it gives us different perspective, and can inspire a lot of research that would not have happened otherwise, because within our Western framework, as I said, we're so much limited by the approach. Of course, developing drugs to treat diseases is very important. I mean, obviously it saves a lot of lives. Once people develop the disease, you need a treatment, and drugs can be life-saving. But at the same time, we want to understand how to not develop the disease. And this is where other types of traditional type medicines can be much more effective, in prevention. And we have to understand, how does that happen? And what are the mechanisms by which these treatments can be preventive? And I think this is where I think the future, and I would encourage the next generation of researchers like yourself to really pursue these goals, because we need it.
Thank you so much, Dr. Longeva, for this inspiring conversation. It's been very, very fun for me. And I hope the audience enjoyed it as much as I did. We are left with so many wonderful, valuable takeaways from today's conversations, and a lot to ponder on. And I feel there is so much more that I would like to explore with you, if given an opportunity. But thank you so much for coming to the podcast.
Well, it's been a pleasure, and I want to wish you the best of luck in your research. And I want to thank you. I think you've got some very interesting perspectives. Your questions were great, and I really enjoyed our conversation.
Thank you.